Welcome to Umina Medical Limited. Ever wondered how the world of science conducts experiments that are too dangerous or unethical to perform on living organisms? Welcome to the fascinating realm of in vitro studies. In a nutshell, the term in vitro translates to in the glass, originating from the Latin language. This phrase refers to a technique in which scientists carry out experiments in a controlled environment, like a test tube or a petri dish, rather than in a living organism or a natural setting. Now you might be wondering, why do we need in vitro studies? Well, there are several reasons. For one, they provide a safe and ethical way to study biological processes. For another, they allow scientists to isolate specific cells or molecules and study them in a controlled environment. This can lead to a better understanding of diseases and the development of new treatments. However, like anything else, in vitro studies have their limitations. Since they are conducted outside a living organism, they may not accurately reflect what happens inside the body. But by using advanced techniques and careful design, scientists can minimize these discrepancies. A common example of an in vitro study is the development of new drugs. Scientists first test these drugs on cells in a petri dish to see if they have the desired effect. If successful, they then move on to animal studies and eventually, clinical trials on humans. Let's not forget about the role of in vitro studies in genetics. They have been instrumental in breakthroughs like cloning and gene editing. Remember Dolly the sheep? She was the first mammal cloned from an adult somatic cell, using the process of nuclear transfer, a prime example of in vitro techniques. But how does one conduct an in vitro study? It begins with the collection of cells, which could be bacterial cells, plant cells, or animal cells. These cells are then cultured in a lab, under conditions that mimic their natural environment. Next, scientists introduce the variable they wish to study, such as a new drug. They then observe the cell's response, which can provide valuable insights into the drug's potential effects on a living organism. In conclusion, in vitro studies are a crucial part of scientific research. They offer a safe and ethical way to study biological processes, contribute to the development of new treatments, and have led to significant breakthroughs in genetics. However, they do have their limitations, and their results should be interpreted with caution. Despite this, they remain an invaluable tool in the scientific community, driving our understanding of biology forward, one petri dish at a time.